So where are we headed today? Well, we started in the introduction last time about talking about prokaryotes and eukaryotes. We didn't really have much time to get down into the details on that. So I'll start the discussion here today with the idea of the distinction between prokaryotes and eukaryotes, but also what they have in common. And one of the threads of our discussion throughout the semester will be this idea of the central dogma. And what we mean by the central dogma is that life on the planet, as far as we know, and it may not always have been this way, but it now uses DNA as a kind of storage molecule, a kind of instruction set, which then, from this DNA, we make RNA. This is a process called transcription. And from the RNA, we make protein. This is a process called translation. We'll go into many of the details about how that's done, but for our purposes today, I just want you to recognize that whether we're talking about prokaryotes or eukaryotes, this idea of the central dogma, in general, broad strokes holds. If you get down into the biochemistry of it, you'll find there are some subtle differences between how the central dogma is carried out in prokaryotes and how it's carried out in eukaryotes, but still, this is the general form of life as we know it. What I want to mostly emphasize here, though, is the difference, the distinctions between eukaryotes and prokaryotes. Prokaryotes were the first of the animals, or first of the creatures, the organisms to come about on the planet, probably 2.5 billion or so years ago. And then sometime between about 2 billion and 1.5 billion years ago, a long time back, a new set of organisms evolved, and we don't know entirely where they came from. But it looks like they probably came perhaps from the archaea, which are relatives of the prokaryotes. But what separated the eukaryotes from both the prokaryotes and the archaea is this idea of compartments. What happened in the generation of eukaryotes is we began to see subcellular functions within the cell, being organized into compartments. And those compartments mostly are going to be membrane bound, and we'll talk about and name those various compartments. But one of the major changes and differences which allowed probably for these eukaryotic cells to come together and work in concert with other eukaryotic cells to form tissues and organs, which prokaryotes never figured out how to do, much of that probably resides in not just the compartmentalization, but the particular compartmentalization of DNA into the nucleus. Well, I'll draw a couple of very simple diagrams that emphasize the idea that in prokaryotes, the DNA is found in the cytoplasm. It's a single ring of DNA. But now, once we begin looking at the complexity of how DNA is organized in eukaryotes, we find that one of the fundamental compartments, the so-called kernel of the eukaryote, is in fact the nucleus, and all of the DNA will be stuffed inside the nucleus. But another important distinction between the eukaryotes and the prokaryotes is that the DNA is organized in a particular way. It's not only stuffed into the nucleus, but that within the nucleus it's organized into chromosomes. In contrast to the prokaryotes, which have pretty much, and maybe a few plasmids, but mostly a single ring of DNA, in eukaryotes, you find multiple chromosomes. In each and every one of our cells, there's a nucleus, and in those nuclei, except for the germ cells, we are going to try to stuff 46 chromosomes, 23 of which came from your father, 23 of which came from your mother, and we're going to have to stuff all of those chromosomes. Several meters of DNA will now have to be stuffed into this compartment called the nucleus. So there have to be some pretty sophisticated organizing molecules which allow for stuffing all that DNA into the nucleus. We're going to see that the molecules which are involved in taking these chromosomes, which are a single strand of DNA, and wrapping them up in an organized fashion, the proteins that are involved with that are known as histones. Histones are a kind of DNA binding protein. What the histones do is they wrap these long strings, these long threads of DNA around themselves, and they form something known as a nucleosome. And these then are the first level of organization of the DNA. It's the histones which allow us to take these great long stretches of DNA and stuff them into the nucleus. But another important characteristic of the nucleus is that although it's forming a compartment, and although it's forming a compartment where the DNA always resides, remember from the central dogma that the RNA is transcribed from DNA. And remember that now, while the DNA is always in the nucleus, the RNA is going to function outside of the nucleus. So that requires a kind of new mechanism. It requires a new structure. And that new structure will be the nuclear pore. The nuclear pore is an opening. It's not just a window. It's a very complicated structure which allows for the movement of molecules both into and out of the nucleus. But for our purposes, it's going to be mostly allowing molecules out. The major molecule to leave the nucleus, of course, is going to be RNA, ribonucleic acid. And we'll talk a little bit about that. One of the important molecules that will be synthesized as RNA inside the nucleus is going to be ribosomal RNA. We're going to go through a whole variety of RNAs. But for now, I just want to remind you that one of the important molecules is going to be ribosomal RNA, and it's made by transcription from the DNA. It then goes out into the cytoplasm. And this ribosomal RNA organizes itself into this particular structure called a ribosome. Now, the ribosome is, in fact, a protein synthesis factory. It's within the ribosomes where we will take RNA generated in the nucleus and translate the information in the RNA into an amino acid sequence and therefore into protein. And we'll see in some detail how that's done. But for today, all I want to say is that ribosomes leave the nucleus, go into the cytoplasm, and in the cytoplasm, they can function to make proteins. At that point, we'll divide ribosomes generally into two categories. Some ribosomes will be free in the cytoplasm. These will be the so-called free ribosomes. But other ribosomes are going to be attached to a membrane-bound compartment called the endoplasmic reticulum. Now, the endoplasmic reticulum is a membrane-bound bag. And one of the ideas of being a reticulum is it's meant to extend throughout the cell. No matter where you go inside the cell, you can find pieces of this reticulum. In some places, you'll find that it's a calcium storage organelle. And in some places, you'll find that its function, as we'll talk about today, is to bind to ribosomes. And what we'll see is that ribosomes, whether they're free ribosomes or whether they're bound to the endoplasmic reticulum, still make proteins. But what we're going to have to recognize is that the free ribosomes make proteins that are fated to become part of the cytoplasm. They are fated to become cytoplasmic proteins. But once you attach the ribosome to the endoplasmic reticulum, that ribosome is now dedicated to making a particular kind of protein. And in general, broad sweeping terms, we're going to see that this rough endoplasmic reticulum, which is derived from the attachment of ribosomes to the membrane-bound bag, will generate secreted proteins, including hormones. They'll make transmembrane proteins, like the ones we talked about last time, the ones that are threaded in and out across the membrane. And they will make a third type of protein called the lysosomal protein. The lysosomal protein is usually an enzyme that will be sequestered within a membrane-bound bag and will serve as a kind of garbage disposal for the cell.